And we are recording. Okay, super. All right, so let's share some screen. Where's A? Right there. Good. Okay, so here we go. So reproductive system, we're going to start with the male. That's the easiest one. Female's a little more involved. In the male, we have the testes, and well, in the male we have the testes, but in the ovary, the the analogous organ is the ovaries. And what we're doing is in those organs is trying to produce what we call gametes or sex cells, and they are haploid, just 23 chromosomes, but we have to use meiosis, right? Two sessions, if you will, of mitosis to create the haploid cells, the gametes that are all different. And so what we do in the to get things going is in the male we have androgens, the most important of which is testosterone, and in the female we have estrogen and progesterone. Right? Okay. So again, the male reproduction, there we go. Okay, now we're rolling here. So the male reproduction, the testes, they are in the scrotum. That's where the sperm production occurs, and it occurs in these little tubules called the seminiferous tubules, actually within the tubule itself. So they're very microscopic. The sperm, of course, is then delivered through a series of ducts to the outside of the body. The epididymis, which we'll show these to you in a minute, the epididymis is where the sperm is stored and develops. And then we start, with the ductus deferens, ejaculatory duct, and out the urethra. There we go. Okay. So let me make sure my thing is here, this pointer. Okay, so here's the testicle here, and this is where the sperm is made in the seminiferous tubules. They then move over to the epididymis here, the epididymis, and they're stored in there until ejaculation, and then they leave here through the ductus deferens, which travels up over the pubic symphysis here, across the bladder, and then opens up a bit here where they call this the ampulla of the ductus deferens, gets larger because it combines with this seminal, it's called the seminal vesicle, which contributes the majority of the seminal fluid here. And then off through this structure here, which is the prostate gland, which also contributes fluid to the seminal fluid. And then down past, now we're down into the penis here and down past what's called the bulbar urethra gland. That gland doesn't really contribute to the seminal fluid. It is the gland that's stimulated during erection, and it secretes a alkaline fluid that lines the urethra to get rid of the acid, essentially, and so that the sperm, when they travel through here, are not destroyed in any way. These glands, the seminal vesicle and the prostate gland, they contribute not just fluid to the seminal fluid, but also antimicrobials and also nutrients, if you will, to supply the sperm with nutrition and energy as they head on their way to the uterus. Okay, so back to the scrotum here. We have paired testes. They have to remain about 3 degrees Celsius lower than core body temperature for optimum sperm production. If they are too hot, then sperm production is not produced. The sperm production is delayed or impaired some way. There is quite a problem with infertility in the United States. It's been going on now for 20 or 30 years. There's a lot of data written about it, collected about studies that have done that indicate that the chemicals in the environment in particular are causing infertility, not only in females, but also in males. with infertility, we tend to blame the female, which is unfortunate because at least half of the infertility problems are also with the male. And it's typically low sperm count. There's in fact, a book I'm reading right now. It's an excellent book. It's written by a scientist who studies this. She's been working on it again for 30 years or more. Take a look. It's called Countdown. It's by this scientist. Her name is, I don't know, I can't find her name. But anyway, the name of the book is Countdown. It's about infertility on the planet Earth. It's pretty amazing. The stats are outrageous. Yeah, anyway. So the scrotum has a smooth muscle that wrinkles up. It's called the dartos musculature. It's smooth muscle because it responds to the environment. If the male is in an environment where the temperature around him is too hot, the smooth muscle relaxes and allows the testicles to get further away from the body to try to stay cool. If the person's in a cold environment, then the smooth muscle of the scrotal sac will contract and bring the testicles up to the body to keep them warmer. There's skeletal muscle involved as well that helps with that movement, and that is the cremasteric muscles that are part of the internal oblique. They assist in that up and down movement as well. So anyway, seminiferous tubules, the site of sperm production. Then from there, we go into the spermatic cord, which encloses not only the ductus deferens, but also nerve fibers, blood vessels, and the lymphatic supply to the testes. We'll take a look at that in a moment. And then within the testicle itself, there are these cells, they call it interstitial cells or Leydig cells. Leydig is a guy's name or lady's name. Identified these cells as the site of testosterone production, which is involved in the production of the sperm. So here we have, someone's got their microphone on. Here's the testicle here and the seminiferous tubules. This is where the sperm is produced. And then as they start to grow, we move them over to the epididymis, which is on the back of the testicle. And here's where they continue to grow until they get, all the typical parts, the tail and the acrosomal head and all that sort of thing. And then this here is the spermatic cord, which has the blood vessels, nerves, lymphatics, and ductus deferens in it. But this is travels up over, the pubic symphysis and away. So what we've done in this picture is we've taken one of these tubules and we've cut through it, and we're looking at it. So we see the lumen of the tubule here. And around here, we see all the sperm in their various developmental stages. As you can imagine, around the very edge here is where we have the stem cells. These are the stem cells where the sperm start to divide from, and they develop as they get further toward the lumen. And then from the lumen, they head into the epididymis. So here again is the bladder. Here the ureter is coming down. Over here is the testicle, the epididymis, and the spermatic cord traveling up and meeting the seminal vesicle here, going through the prostate gland and down into the urethra. In the penis, we see that this is the bulbar urethral gland. In the penis, we see cutting through here, looking at it, there's a cross section here. We've got two large bodies on top called corpora, or body, cavernosum, large cavernous bodies, and in the center of which there's an artery. And these are the cavities here, these the blood vessel supplies all of this tissue, and this tissue here is what maintains the erection in the male. The one down below that is the corpus spongiosum, which simply maintains the patency of the urethra. so the sperm can come out there. Okay, sperm production or spermatogenesis. What we're trying to do here is produce a one-end or haploid sperm. You know that all body cells are diploid, right? Two-end. 23 pairs of chromosomes, homologous chromosomes. 23 from mom and 23 from dad. They're creating 46 chromosomes per cell. What we need to do is to create a gamete that has only 23 chromosomes in it, the haploid number, but it can't be exactly, they can't be exactly alike. So we've got to, mix up the chromosomes a little bit, mix up the genes a little bit in the chromosomes. So to do that, we undergo meiosis. So again, we start with the stem cell called the spermatogonia. It undergoes this nuclear division, mitosis, and then one of the cells will stay behind, to continue to produce, continue to divide mitotically. The other cell that was produced will leave and undergo this process of spermatogenesis. And so what we'll ultimately do is end up producing 4 sperm from this one stem cell. Again, they're not identical, but there are 4 of them. In the female, what we end up with is 3 what we call polar bodies and 1 ova, 1 viable egg. And the way that we mix it up so that none of the sperm are alike is through a process called recombination or crossover during prophase 1 of meiosis. The homologous chromosomes get together, again, the one from mom, one from dad, and they exchange genetic material. And then they all split again, and so you end up with 4 different 4 different sperm with different genetic makeup. So here we are, prophase 1, and you can see that they are starting to line up here. And, we've already gone through part of the mitotic event where, the chromosome 1, say, for instance, chromosome 1, there's the green one from mom and the purple one from dad. And what they've done, they've already reproduced, copied each other. So you've got 2 of chromosome 1 from mom, 2 of chromosome 1 from dad. And, we've developed these sister chromatids. But the homologous chromosome, not the sister, not the exact duplicate of, say, chromosome 1 from mom, but the homologous chromosome from, like, dad, they get together, the 2 homologs get together, and they do this little crossover thing here. And so you can't see it very well, but they're showing you that a section of genetic material is now on the chromosome from mom, supplied by dad, and vice versa. And then, of course, we get through metaphase 1 and anaphase, telophase. And then following those events, we go into meiosis 2. Now we're taking these, these these homologous sister chromosomes here, and we're then going to split them up again. And so we end up with, 4 sperm, each haploid number, only 23 chromosomes, and they are all different at the end. So we have, this looks like a whole lot of stuff from mom. This is mom and a little bit of dad. This is dad and a little bit of mom, and this is all dad. And that's how we do it. And each time we produce a sperm, there is different crossover, different genetic material exchanged. And ideally, we have, no 2 sperm alike. And the same thing happens in the female. And when you put those 2 together, you certainly, there's no way in the world you're going to get 2 people that are exactly alike genetically unless you've got, identical twins. So this is we're looking at that seminiferous tubule again, where the sperm are being produced within the testes. here are the stem cells, the spermatogonia here. And you can see that they're going, this is the one that is, this underwent mitosis. And now we have the one of those mitotic cells that's going to continue along and create a sperm. This one stays behind and continues to, be the stem cell to create the line of, sperm. So we end up undergoing, meiosis 1 and then meiosis 2 with 4 sperm, and then they start to, try to develop, and they end up going into the lumen of the seminiferous tubule and then off to the epididymis, where they continue to grow and get ready for their big event. Now you can see that these cells are sitting in this yellowish mass here. These purple things are the nucleus of what we call the sustentacular cell or Sertoli cell. Again, we have 2 names for everything. So sustentacular cell or Sertoli cell. Those cells are what help create fluid that keeps everything moving along. They also are the, keep in mind this is occurring in the male puberty. So we're talking about, something that's occurring around between, I don't know, 10, 11, 12 years old. So not 10, like 11 to 13 years old, 11 to 13 years old. So these guys here, these sperm are not recognized by the immune system. So the sustentacular cells have to create this barrier, between, so we call this the testes blood barrier. So it keeps the immune system away. They also are a bit phagocytic, and they, take care of anything that might be laying around that needs to be taken care of, destroyed, and they supply some nutrients. So getting back to the sperm, the major regions are the head, where the genetic material is. That's where the nucleus is. And then above that, there is the chromosome. I'm sorry, which has the hydrolytic enzymes in it. Acrosomal head, and that has hydrolytic enzymes. So when it gets, to the oocyte, and there's a covering around the oocyte, it has to burn through there, and that's when the acrosomal head is released with these hydrolytic enzymes. Behind that is where the nucleus is. And of course, then behind that, we have this mid piece, which is full of mitochondria that produce ATP, so we can, keep the flagellum moving here, the tail. So we have them starting out here as very young sperm and ultimately developing the tail, and the acrosomal head here with the enzymes. Behind it is the nucleus with the DNA, 23 chromosomes. And we also have the section here, which is full of mitochondria to produce ATP, which keeps the, the flagella moving. Again, here's sustentacular cells, Sertoli cells. Again, they're the blood testes barrier. They provide nutrients and signals for the dividing cell. They dispose of excessive cytoplasm, and they secrete that fluid that keeps the sperm moving through the testicle toward the lumen of the seminiferous tubule. All right, so that was that. Let me get to the second set of slides. There we go. And where's the second set? I don't know. Give me a second. I'll be right back. Let me shrink this down and open these up. All right, so we took care of that. Close that. Bring another one up. Bring another one up. Put that up. Down, down, and up. Good golly, Miss Molly. Give me a second here. I don't know what's going on. There I am. Sorry, I was stumbling around a bit. let's share this second set of slides. Okay, now we're moving forward. So again, as I'm sure you've realized, like everything else in the body, the hypothalamus is involved here with spermatogenesis. And what the hypothalamus does is it releases the gonadotropin releasing hormone around the time of puberty, between 10 and 13 years old in males. And the gonadotropin releasing hormone goes down to the anterior pituitary and then causes the release of follicle stimulating hormone and luteinizing hormone. You know, the follicle stimulating hormone will stimulate the stem cells, the spermatogonia, to start, you know, the process of producing sperm. And the luteinizing hormone is going down there to get that, you know, testosterone produced from the interstitial cells or the Leydig cells. Okay. So again, testosterone is, you know, the final trigger for spermatogenesis. There's also a negative feedback loop to the hypothalamus so that, you know, sperm production isn't overproduced. And the hormone inhibin is involved in that. But testosterone also, as you know, produces the male pattern development, you know, male sex organs, muscle tissue, skeletal growth, skin thickening. The larynx around the age of 10 to 13 becomes a little enlarged. The boy becomes, gets a deeper voice. And also is responsible for the libido or sex drive in the males. So that was it on the males, pretty simple stuff. Females, we have quite a bit to talk about. So the female reproductive system involves not only the anatomy, but 2 cycles that are typically performed within a 28-day period. We have the ovarian cycle, what takes place inside the ovary, and then we have the uterine cycle, what takes place concomitantly in the uterus during that 28-day cycle. And all of those things are regulated by hormones. And the primary hormones, of course, as you know, is estrogen and progesterone. So let's look a little bit of look at a little bit of the anatomy. The ovaries are the female gonads. Here is where the gametes are produced. You know, the ova or oocytes. Again, they're 23 chromosomes or 1N or haploid. The 2 hormones are estrogen, progesterone. They are secreted by the developing oocytes, as well as a number of other tissues we'll talk about in a moment. So the ducts that we know, we need some ducts to get things around here. We have the uterine tube, the uterus, the vagina. Let me see, hold on a second. We did that. So here we are. Here we have the opening to the vagina here. This is the Bartholin's gland here, which is similar analogously to the bulbar urethral gland in the male. It's for lubrication primarily. Clitoris is here. Bladder here. Urethra is here. So vagina going up here, and we have the cervix, which is the opening to the uterus. This is the uterus opening here. You can see all the musculature here around the uterus. And then up here, we have the uterine tube or fallopian tube, as some people call it still. And it comes down, connects to the top of the uterus. And at this end, we have the, excuse me, the infundibulum here and the fimbriae of the uterine tube here, which create a current, like little fingers. They create a current here that sweeps up the oocyte when it is ovulated. And this is the ovary here. Okay, so let's take another look at this. So here we are looking at both ovaries. right and left. We have the uterus here. You can see the cervix, vagina here, muscle tissue all around. So here we have, we'll get a better look at this in a moment, but this is where the oocytes are developing. The oocytes actually start development with their follicle covering in the fetal development, when the child is in fetal development. Then it stops and doesn't start again until puberty. So here is where ovulation occurs, and the oocyte is swept up by the fimbriae here, creating that current, and then starts to travel up the uterine tube here on its way to the uterus. And it's somewhere in here, you know, the ampulla, infundibulum. This is typically where the sperm contacts the egg and fertilization occurs. So those sperm with their, you know, with their flagella, they have, you know, quite a trip. They've got to get into the vagina and then through the cervix, and then through the uterus, and then along the, you know, uterine tube all the way to this spot. Sometimes after fertilization, that fertilized ova, now called a zygote, doesn't get all the way back to the uterus. Sometimes it gets stuck in the uterine tube and starts to develop and divide. The cells start dividing, and we get what's called an ectopic pregnancy. That's very extremely painful. And they've got to do the best they can to get it out of there, or take off that entire section, ovary and tube as well. These are all the, you know, ligaments that we don't need to get into. We won't get into. They have names to them to keep, you know, the ovary up against the abdominal wall and all that kind of stuff. So let's move along. So in the ovary, we have a follicle that surrounds, that's a tissue that surrounds the egg. Initially, it's, again, only one cell layer thick, and then, but it grows. So the follicle surrounds the oocyte here, and as it grows, just before ovulation, we call it the Graafian follicle or mature follicle. It is filled with fluid in one section. And at ovulation, what is ovulated is the oocyte, the egg, surrounded by some tissue like the corona radiata and some tissue like that. But the follicle itself remains behind in the ovary and turns into what's called the corpus or body luteum, corpus luteum, or luteum means yellow in, I guess, Latin or something. So it's called the yellow body. It remains behind in the follicle and becomes a hormone producing tissue. So here we are in an ovary. And here we are starting off with these primary follicles. These follicles were created in utero during the fetal development, and they stop the development in prophase 1 of meiosis 1, and they will remain there, giving off a little bit of estrogen. The center of this is a oocyte. The follicles are surrounding the oocyte, and the follicles are producing a little bit of estrogen throughout the child's life, up to and including puberty. So following puberty, we get an increase in estrogen, and then we get one dominant follicle with its oocyte in the center, and it continues to grow and grow. So the follicle is getting larger, producing a lot more estrogen. And we develop this at some point here, this very large Graafian follicle, which is the mature follicle, which the artist chose to make it look like a big happy face with the two eyes, the nose, and the big mouth. But ultimately, it gets to a point, say, you know, day 14 of a 28-day cycle, and ovulation occurs. So the ovulated oocyte takes off, swept up by the fimbriae into the uterine tube, and what's left behind here is the follicular tissue here. Now, it then starts to develop into the corpus luteum. And this is a fully developed corpus luteum that will produce estrogen, progesterone, inhibin, and relaxin while we're waiting for this little guy here, ovulated oocyte, to become fertilized and implant. Okay. So again, the uterine tubes or fallopian tubes, the expansion at the end is called the infundibulum. This is typically where the fertilization occurs. The ciliated fimbriae at the end of the tube sweep up the oocyte into the fallopian tube or uterine tube. The uterine wall of the uterus has 3 layers. The perimetrium is the outer layer. It's attached to the visceral peritoneum. The myometrium is the largest layer. This is the smooth muscle tissue in there, and the endometrium is a mucosal lining, which we'll talk about in a few minutes. Right now, in fact, the endometrium has 2 layers to it: the stratum basalis, or the basal layer, which doesn't change much, except for the fact that here is where we have mitosis of the cells that will develop the functional layer. And the functional layer is the layer that they also call the stratum functionalis. That is the layer inside the uterus that is replete with, nutrients and blood and all kinds of, glycogen and fluids and everything that any fertilized ova would love to land on to implant to start to develop. So that's the stratum functionalis. It's developed over that, that 14-day getting to the ovulation, but then continues to develop after that up until menstruation if there's no implantation. So this is the layer, the stratum functionalis, that is shed during menstruation. Sometimes they call it the deciduous layer. Deciduous meaning just like trees. Deciduous trees are the trees that have the leaves that fall off in the autumn. So sometimes they call it the deciduous layer. Uterine vascular supply, the uterine arteries come off the internal iliacs, then they become the arcuate. And the ones that I really want to remember are in the radial branches. The ones I really want to remember are the straight arteries and the spiral arteries. These are the ones inside the uterus that create this environment. Straight arteries supply the base layer, the basalis layer, where the mitosis occurs. The spiral arteries grow into and create the stratum functionalis. The spiral arteries are the ones that, if estrogen and progesterone are no longer available, these are the ones that kink up and break down and bleed and give rise to the stratum functionalis, leaving the uterus as the menstrual flow. So here is the muscular layer of the uterus here. As we come up, we have some straight arteries here, and then they go spiral. And these spiral arteries grow and give rise to all these other vessels here that create this functional layer, this deciduous layer, that's growing, each month, awaiting the implantation. And then these guys are also created too, these uterine glands that contribute to the fluid. So oogenesis, we're trying to create the female gametes. It begins in the fetal period of development. We have these stem cells called oogonia, which are 2N, just like spermatogonia. I'm sure I made that clear. They're also 2N. They are diploid. They have they have 46 chromosomes, 23 pairs, one from mom, one from, 23 from mom, 23 from dad. So these stem cells then undergo mitosis and ultimately form these primary oocytes that will start meiosis. And again, we're still in the fetal period of development. And the meiosis begins but then stops in prophase 1, and they stay there until puberty. Okay, so again, throughout childhood, the ovaries and the follicles are producing small amounts of estrogen. And that estrogen is inhibiting the hypothalamus from releasing the gonadotropin releasing hormone. And as well, as long as the estrogen is being produced, the hypothalamus will not release that gonadotropin releasing hormone, which essentially will start, the process of, reproduction, the female reproductive cycle. So as we get close to puberty, the adipose tissue starts to accumulate on the female, and the hormone leptin is released. And what leptin does is it makes that hypothalamus a little less sensitive to that negative effect of estrogen. And then all of a sudden, the hypothalamus releases gonadotropin releasing hormone again at puberty. And we're talking like 11 years old or so in the female. And the GnRH causes the release from the anterior pituitary of follicle stimulating hormone and luteinizing hormone. So ultimately, after about 4 years of this occurring on a monthly cycle, we end up with this regular 28-day cyclic pattern. Not everyone is 28 days. Some people are, 26 or a little longer. And even the established pattern can change, within one person. And then, menarche occurs. Menarche is the first time that the female has bleeding or the loss of that functional layer. Okay, oogenesis. Again, at puberty, each month, we have a few of the oocytes, these primary oocytes that we showed you earlier, start to get activated. They're producing estrogen and progesterone, and that is allowing one of the follicles, now we call the dominant follicle, to develop on its own and it starts the meiotic pattern, meiosis occurring again. And it will continue until we get to metaphase 2, and then it will stay at metaphase 2 and be ovulated at metaphase 2. So again, the increase in estrogen progesterone causes this one single dominant follicle to be stimulated, to go back into meiosis, move through meiosis 1 into meiosis 2, but stop at metaphase 2. And then that's what is ovulated, the oocyte in metaphase 2. So at this point, we're calling it a secondary oocyte, and it is typically ovulated on day 14. And if fertilized by a sperm, it completes at that point, that stimulus of fertilization completes meiosis 2. And we now, at the time of fertilization, we call that cell, the first cell of the human body, we call it a zygote because it now has 46 chromosomes, now has 46 chromosomes, 23 pairs, one pair, 23 from mom, 23 from dad. So we're going to talk about the ovarian cycle, and we will also talk about the uterine cycle. So the ovarian cycle, give me a second. Okay. The ovarian cycle is in 2 phases. The follicular phase, which is the time that we're, the follicle is growing, day 1 to 14. So that follicle is that tissue around the oocyte. It's growing larger and larger and producing lots of estrogen and progesterone. Then at day 14, we have ovulation, mid-cycle. And then we have the second phase of the ovarian cycle called a luteal phase. Luteal, meaning that we're going to create the corpus luteum, and that's day 14 to 28. Then the corpus luteum releases, produces and releases estrogen, progesterone, and all that to, essentially keep the uterus, growing that functional layer. Okay. So again, here we are. We're just showing you a few things more. We've seen this before. This is the follicles here, primary follicles, then moving into secondary follicles. That there's one dominant follicle that, again, within it has the oocyte. And the oocyte, the follicle gets larger and larger until it's called the Graafian follicle. And then at day 14, we have ovulation. Okay. So ovulation. Ovulation occurs, the release of the oocyte occurs at day 14 because of a surge of luteinizing hormone. That's what softens and causes the ovarian wall to rupture, releasing that oocyte. Sometimes people, express that they can feel that little twinge of pain, and we call that Mittelschmer's sensation. Sometimes 1 to 2% of ovulations, we get more than one secondary oocyte released, and if both are fertilized or several are fertilized, they're referred to as fraternal, twins or triplets or quadruplets. Identical twins, which means that we have, two people that are identical in genetic makeup, that occurs after fertilization. So we have one oocyte that's fertilized, we have one zygote, everything's going fine. And then for some reason, that fertilized zygote decides it wants to undergo mitosis. And, mitosis, we get two cells exactly alike. And that's called, identical twins. All right, then. We're just showing you a pretty picture of ovulation. That's all we're doing here. The luteal phase, again, that Graafian follicle collapses and becomes a corpus luteum, and the corpus luteum remains in the ovary, and it creates relaxin, inhibin, and progesterone and estrogen. And again, if no fertilization occurs, then of course we don't need the corpus luteum anymore, and it turns into the corpus albicans or white body in about 10 days or so. If we do get fertilization and pregnancy, then we have the corpus luteum remaining there, being a hormone-producing tissue now, until the placenta takes over. And we've got to build the placenta, which we'll talk about in the next chapter. We're going to build a placenta, which will then produce all of those, estrogen, progesterone, relaxin, and inhibin. But until then, it takes about 3 months to build a placenta. Until then, the corpus luteum takes care of all of that. So what keeps the corpus luteum alive? Believe it or not, it's human chorionic gonadotropin, which is coming from the developing zygote. That fertilized egg, now called a zygote, is the place where we get the human chorionic gonadotropin released from, and it goes back up and keeps that corpus luteum functional. And we keep showing you the same thing over and over again, but this is actually showing a good picture of the corpus luteum fully developed here. All right, so that's done there. We've got those slides completed. And we have one more set of slides, and I think we'll be done. What time is it? 6:45? Yeah, we're doing good. We'll do the rest of the slides if I can get there. If you have any questions, let me know, of course. Just unmute your mic and yell them out. All right, so we're going to talk a little bit about those hormonal interactions over that 28-day ovarian. Again, we're in the ovarian cycle. Again, day 1, we have gonadotropin releasing hormone causing the release of follicle stimulating and luteinizing hormone. And what they're doing is they're going down to the ovary and they're trying to get things going. They're trying to get the follicle stimulated so that we can develop at least one primary follicle that's going to move forward. And the luteinizing hormone is going down there to start, the estrogen release from these developing follicles. Estrogen levels are rising. And as estrogen levels are rising from these developing follicles, what they're doing is they're going back up to the hypothalamus, and they're, again, inhibiting the hypothalamus from releasing that gonadotropin releasing hormone and the FSH and LH. So it's those hormones are sitting there. Actually, they're not sitting there. What's happening in the anterior pituitary is FSH and LH are still being created, but they're being stored, and they're getting larger and larger and larger in amount. And so the anterior pituitary was synthesizing these hormones, but they're just sitting there until about day 14. And as this dominant follicle continues to grow, it's releasing more and more and more estrogen, and then all of a sudden that much estrogen in the bloodstream reaches what we call a critical point and now is no longer inhibitory, but becomes a positive factor. And we get this rush from the anterior pituitary of luteinizing hormone on day 14. And it's that flood of luteinizing hormone on day 14 that softens up the ovarian wall and causes ovulation. So, where are we? Luteinizing hormone. So it causes ovulation, does a couple other things too. It causes the oocyte to move forward, the egg to move forward out of meiosis 1 and into meiosis 2 until it gets to metaphase 2. And we can call it a secondary oocyte, which is, what is ovulated on day 14. It triggers ovulation, as we have said. And it also helps with stimulating the Graafian follicle to turn into the corpus luteum. And then the corpus luteum is maintained by the human chorionic gonadotropin if indeed there is fertilization. So again, the corpus luteum produces relaxin, inhibin, progesterone, and estrogen. Inhibin, progesterone, and estrogen inhibit any further release of FSH and LH because we don't want any more ovulations occurring, while we're trying to get this secondary oocyte fertilized and implanted and go through, a pregnancy. So those hormones, which again are first released by the corpus luteum and later by the placenta 3 months later or so, we want to stop that LH and FSH, from developing. From being released. Okay, again, fertilization. If we get fertilization, the zygote, now we have this little cell, single cell, with 46 chromosomes, starts to produce human chorionic gonadotropin. And that's what keeps the corpus luteum alive. If there's no fertilization, then there's no HCG and the corpus luteum just degenerates. Okay, and then of course that stops the blockade to FSH and LH, and then we start the cycle all over again. And again, no fertilization, no HCG, no blockade on FSH, LH, and so we get right through to the end of the 28 days, and on day 1, since we no longer have a nice production of estrogen and progesterone, we lose the functional layer. And that's the beginning of the first phase of the uterine cycle called menstruation. So it's just a little graph here. This is kind of cool in your book. Please take a look at this. You can look at the slides as well, but look in your book, your text, and you'll see on one particular page, I forget what page it is, but you'll see these charts one above the other. And it's showing you that 28-day cycle from day 1 to day 28. And the colors keep you lined up. So here we are at like day 14, luteinizing hormone is at its peak and is released, and as well as FSH, excuse me. And this is where we get ovulation from that flood of LH. Here we have, again, you watch the colors, day 14, we get ovulation. Until that point, day 1 through 14, we're, growing this follicle around this oocyte. Then we have ovulation. Oocyte, secondary oocyte leaves, and we're left behind with the follicle that develops into the corpus luteum. And then if we don't get, HCG, then it just degenerates up until, day 28. This shows you the estrogen and progesterone levels as they are, they're rising from the developing follicle around the oocyte until we get this peak of estrogens around day 14 that then, causes the release of LH. Up until that point, it was causing the sequestering of LH and FSH. When it gets this high, it creates a critical level and causes the release of those two hormones. And down here, now we're looking at the that same 28-day cycle, but we're looking inside the uterus. And this is the basal layer down here, doesn't change, but it is mitotic. And so in the beginning of the phase, we have the 1 through 5 menstrual flow here. That means that we didn't get fertilization. And so as we move forward from day 5, however, the follicles are beginning to develop again and getting large and releasing estrogen and progesterone. And that's coming down here and causing these spiral arteries to grow in and this nice functional layer to develop. And here we're at day 14. And so we continue to develop this functional layer so it gets full of nutrients. And so we have this beginning again, 1 through 5 of the uterine cycle is the menstrual phase. And then we have the proliferative phase, proliferative referring to the deciduous layer here or functional layer up to day 14. And then after that, secretory phase occurs. So there's 3 phases in the uterine cycle. Secretory phase, and that is where we get lots and lots of nutrients and fluids, flush with blood and ready to receive that fertilized egg. Okay, this is what we just talked about. So again, 3 phases, uterine cycle, changes in the endometrium over 28 days. 1 through 5 is menstrual, 6 through 14, and we call proliferative. It is the pre-ovulatory, in other words, prior to ovulation, right? And then days 15 to 28 is secretory, lots of secretions, building up in the functional layer. And that's post-ovulatory, obviously. All right. Uterine cycle, again, we're just going over the same thing again, but you know what's going on here. So menstrual phase 1 through 5, the functional layer of the endometrium is shed. And that's what creates the menstrual flow. In the ovary, the hormones are at their lowest. That's why we got the breaking down, kinking, if you will, of the spiral arteries that are supposed to, maintain that functional layer. So again, estrogen, progesterone are low. There's no HCG, no fertilization. And also around that time, we're still going to back up in the ovary. We're still getting these follicles wrapped around the oocytes starting to develop. And so we see a rise in LH and FSH, from the pituitary. No, those 2 sentences didn't go together. Around the same time, the follicles are developing and they are beginning to produce more estrogen, right? More estrogen, progesterone. And that estrogen, progesterone is causing the anterior pituitary to sequester this, its creation anyway, of luteinizing hormone and FSH. So that's starting to build up again in the anterior pituitary, those 2 guys. All right. So then there's the proliferative phase, which is the, again, before ovulation, pre-ovulatory, days 6 to 14. This is where the estrogen levels are rising. The ovarian follicles are in the ovarian follicles, as the endometrial functional layer is developing, spiral arteries are growing. It is also considered the most variable. In other words, we say it's day 6 to 14, but that doesn't mean it truly is. So people that are trying to not get pregnant and using these natural rhythm cycle, if you will, this is where they get messed up, because day 6 to 14 in the proliferative phase isn't always exactly 6 to 14. So it's most variable in length. Again, the glands are enlarging, spiral arteries are growing, and then ovulation occurs on day 14 because of the luteinizing hormone surge. Following that, post-ovulatory, day 15 to 28, which is constant, when we finally get ovulation, the next days, whatever day they are, 15 to 28 or whatever, are extremely constant, time-wise anyway. And the corpus luteum is developing, the progesterone is helping the spiral arteries to grow, stratum functional layer is growing, glycogen's accumulating. And this is the point at which that post-ovulatory secretory phase, where we develop what's called the mucus plug in the cervical area of the uterus. So the uterus is no longer letting anything in. So just showing you that again, estrogen rising day 14, et cetera. And here's progesterone coming up. This is where we're showing you that getting a rise in progesterone is helping the spiral arteries, grow like crazy into the functional layer to create a big, thick functional layer. Right there in the green. So again, post-ovulatory, we have the secretory phase of the functional layer. If fertilization does not occur, of course, post-ovulation, then the corpus luteum degenerates, progesterone levels fall, spiral arteries spasm and die, they constrict, and so you no longer have any blood supply to the functional layer, and we have menstrual flow on day one. We're back to day one again. Estrogens, so we're done with that. Estrogens, the hormone, there are actually three of them. We talked about them collectively as estrogens. There's estrone, estradiol, and estriol. They clearly are what's promoting oogenesis, follicle growth. They have anabolic effects on the, growing effects, obviously, on the female reproductive tract. They induce those secondary sex characteristics like breast growth, subcutaneous fat, widening and lightening of the pelvis. There are more metabolic effects again, prior to menopause. They help maintain a normal cholesterol level and high HDL. Remember, HDLs are the good, if you will, cholesterols that, bring excess cholesterol to the liver to get rid of it through the bile. Postmenopausal, hold on a second. Again, estrogens in premenopausal anyway, facilitate calcium uptake, for muscle contraction and strong bones. And so when we look at the menopausal woman, the postmenopausal, we see sometimes, a loss, or not a loss, but a significant decrease in estrogen. And so we have maybe higher cholesterol levels, lower HDL levels, and they start to lose, calcium uptake into the bones, and, it's more easily, something like osteoporosis is more easily developed. There's another problem. Menopause clearly is not pathological, but female athlete triad is a bit, if you will, pathological. It's really an eating disorder. And what happens is you get these young ladies in their like 20s, late teens, early 20s, or mid 20s perhaps, and they are obsessed with exercise, and they are obsessed with their bodies. And so they don't eat properly, and they exercise a lot. And what they end up with is a significant decrease in body fat. And again, if you've got decrease in body fat to such an extent that you're not producing adequate amounts of leptin, then what you end up with is a decrease in estrogen and progesterone, and essentially amenorrhea. And then if you have amenorrhea, and again, decrease in estrogen and progesterone, you end up with a decrease in bone density. So we get these young ladies who are amenorrheic and have a bone density that looks osteoporotic. One more slide. So again, menopause, declining estrogen levels, we end up with atrophy of the reproductive organs and breasts, sometimes irritability and depression. We get hot flashes from unusual vasodilation of the superficial blood vessels in the skin. And so these people say that these women say that they have, they get this outrageous sensation of heat all over their body from intense vasodilation. The skin thins out a little bit, and of course, bone loss, and again, increased cholesterol levels and decreasing HDL levels. All right, so that's that. All right, so we're done with that. We managed to finish that chapter. That's reproduction. So please read the lecture notes on that. There's quite a few. We have the chapter notes. We have notes that are just on the female reproduction. So please take a look at those. The one thing we didn't talk about is teratogens, which I'll talk about right now because I just see it in my paperwork here. We didn't have them in the slides for some reason, but they're very important. Teratogens are a substance, any substance in fact, that might produce physical or functional defects in the human embryo or fetus. Teratogens can cause physical malformations, problems in behavioral or emotional development, decrease IQ, anything that complicates the pregnancy, such as preterm labor, spontaneous abortion, miscarriages. There's essentially 4 types of teratogens. There are physical agents like ionizing radiation, such as X-ray, hyperthermia, elevated blood temperature. That means over, of the female, the pregnant woman, over 102. There's metabolic teratogens like malnutrition, diabetes, thyroid disorders. There's teratogens from infections, rubella virus, herpes simplex virus, syphilis. Also, there's drugs and chemicals, and like alcohol, cocaine, thalidomide, etc. So a good example again is the thalidomide back in the 1950s. I think it was 1950s, 1940s or 50s.
Question.
Hold on. Let me tell you this one thing. Thalidomide was a medication that was released in Europe, not in this country, in Europe, for morning sickness. And so they released it, and women took it who were pregnant, and morning sickness always comes early, like at the beginning of pregnancy when you're in the embryonic phase. Embryonic phase is the first 8 weeks. After the first 8 weeks, we then call it the fetal phase, fetal development. So in the embryonic phase is when we're developing, like the limb buds, arms and legs. And unfortunately, the thalidomide in some women who took it, it caused the limb buds not to develop. And so babies were born with no arms or legs. And that is probably the most intense example of a teratogen that's for recent times. But yes, go ahead with your question.
Can antibiotic affect pregnancies, like in the early stages?
I don't know of any that can, but some of the antibiotics they have now are extremely strong, and I wouldn't doubt that they could cause some problems because we have bacteria now that are resistant to antibiotics, and so they're making them stronger and stronger. Yeah, I'll just stop there. I mean, the place to go for that kind of information is that website I always tell you about, the Environmental Working Group, EWG. Please look them up and just go to their website, go into the search box and type in, antibiotics /pregnancy. And if there's something about that, I'm sure they've studied it, because they study all of the chemicals on the planet and how they affect humans. They're really big on that. They're really big on that book I mentioned, Countdown. They do those kinds of studies. How these chemicals, like the chemicals we find in plastics, how they affect development, how they affect fertility. They're finding really interesting stuff. Like believe it or not, one of the studies that they have done, or they've been doing for years now, is measuring, I know it sounds bizarre, but they measure the distance between the anus and the genitals. And the distance between the anus and genitals in women, females, is significantly shorter than in males. But what they've been finding over the past 30 years or more is that distance is decreasing in males. And so they are becoming, I hate to use this word, but reproductively anyway, they're becoming more feminized, and their sperm counts are dropping dramatically. In fact, the sperm count of the average male today is about half of what it was 50 years ago or more. So, and that's from chemicals. That's from exposure to the, PCBs and all those plastics and stuff like that. Could antibiotics affect a pregnancy? I'm sure they can, but I don't know. I mean, they certainly affect all of your normal microbiota, the normal bacteria that you have in your body that live with you and keep you alive and keep you healthy. And, the more they research that, the normal bacteria in your gut, the more they realize how important that normal bacteria is. And taking any antibiotic, destroys a great deal of that, so much so that people end up with diarrhea, and you're told to eat yogurt when you're taking your antibiotics. And so, how much of the normal microbiota that keeps you healthy all day actually helps in pregnancy? Who knows? I don't know. So yeah, I mean, I'm sure this study is going on about it right now. Any other questions?
No, sir. Thank you.
Anybody else? No.
No, thank you.
All right, we're good for tonight then. On Thursday, we will do the fetal pig lab. It wouldn't take a long time, but we will do that, and that will be the lab and the review for the upcoming practical 3. All right. Well, thank you for coming tonight. It's good to see you all, and have a good evening. I'll see you on Thursday. Thursday.
you. Have a good night.
Thank you. Have a good night.
Thank you.
Thank you.
Good night.
Good night.
Thank you. Have a good night.
Thank you.